Okay, so we'll go ahead and get started here. So from last week, what is some of the bigger concepts we had talked about? For instance, what's a purine? It's different than a pyrimidine, because we even read about that. So a purine would be which two nucleotides? Why are you right?
Because
Other than, and you could say it says that in the book, and I would say that that's right, or it's because that's what we told you it would be. So if a purine is adenine and guanine, well then a pyrimidine must be cytosine and thymine. Okay? Basically the reason is because that's what we just told you it was. And we could reinforce that by reading in the book like what we did. For individuals who are choosing to follow that, okay? So then when it comes to nucleotides, how do you know which nucleotide bonds with each one? Because there's a pattern that we told you. What would that possibly be?
adenine with guanine.
Nope, then you're saying that's a purine, where those are both purines, but they don't bond with each other, though. So what does adenine bond to?
The guanine.
No, not guanine. Adenine bonds to what? Bonds to thymine. So then what does guanine bond to? Cytosine. That's right. And I don't know if that's what you said. I heard some commotion. That's the problem when you get older. We probably hear things when we're trying to pay attention, and if we say something inappropriate, well then, they can't hear us. But we hear that, but yeah. Anyway, this is where we left off with. So purine bases, like we just said, adenine and guanine, pyrimidine, cytosine and thymine. So what does that mean? Well, that adenine bonds to thymine, and cytosine bonds to guanine. So you could say that these two bond together, and then those two bond together. Okay, that's something that we had done last week, and we also put a strand of DNA up here. How would you know that, other than us telling you that this was a strand of DNA? It had 3 circles on here. What's the biggest reason that we would know that this was a molecule of DNA? And here's your clue right here. We said something about, say that again? Yeah, twisted ladder. So it's got a double helix. This is one half the helix. This is the other half of the helix. Hence, one on each side, a double helix, okay? Which is going to be different than another type of structure, because if we have a double helix, we must have something that is only a single helix structure, and that would be correct. Okay. Oops, wrong button. So this is where we begin. The replication of DNA. So we had shown you this on Friday. We showed you the process of replication, but we left out the enzymes that cause this. Well, I don't know if you want to say cause it, but maybe more so allows it to happen. Because keep in mind, tomorrow we'll have an activity in here, but we would like to get through this content Otherwise, it might not make any sense. And maybe for some individuals who try to accomplish this or perform this activity, we might not know what we're doing. And maybe that's by choice. I don't have an answer for that. But in other words, let's see here. Because I was giving the volleyball player, not a hard time, but I was just saying, you should just look across at your opponent and just say, I'm going to jump as high as an expanded octet. Now look at them and, wow, what are they talking about? They must be smart, which they are. Okay? So then we could also say, well, okay, why do you get better at volleyball or cross country? How do you get better?
Work hard.
Yeah, you work hard, and what's another way? It's not just working hard. Sometimes some people do work hard. We can't deny that. And sometimes we just don't get it. Okay?
Natural talent.
And there could be natural talent. That's true. But then what is the best way to try to improve at this, whether it's in class or for athletics? Practice. Do your best. I mean, I think that's pretty elemental, don't we think? I mean, if we're going through this, well, there's hopefully a reason. I would like to say there's always a reason, but with this, if we don't go through this, what we're going to be doing tomorrow just won't make any sense. Okay. So we said this last week. We said something about, okay, scars or wounds or something to that extent. Okay? When you need to make new cells really quickly, okay, those cells have to have the same function. And we know what is it that you find in each cell of our body. Besides just a nucleus, it's something that's in the nucleus. What does the nucleus do?
It gives directions to the cell.
Exactly. It gives directions to cells. But what are those directions coded on something?
DNA.
It's coded on the DNA. That's right. Okay? So, let's see here. Lost. Where is it, my presenter? Got up and walked away. Okay, well, I think we just had this one left anyway. Nope, I didn't. There we go. So we will put to words, we will make the diagram according to the words that we see up here. Oh, here it is. It was hiding. Okay, does everyone have this content that's choosing to write it down? Okay, are we good? Okay. So go ahead and shut this off. Okay. So now, should we not write this down? Should we not, but not be listening, whatever. Now is the chance that you can actually participate and help us out here. Okay? So what I mean by that, okay. What do you suppose is one of 4 nucleotides? What's the only 4 nucleotides we've talked about? Because really there's 5.
Adenine,
That's 1.
guanine,
That's 2. Thymine. You're starting to say it with a C. Cytosine. Okay? So then, of those 4, you said adenine first. Then you said guanine. Then someone said thymine. And then finally, last one was cytosine. So those are our 4 choices of nucleotides. Okay? So therefore, of those 4, start picking out a code that goes in here. Of any one of those 4. Okay? Next. Next. Or you just want me to make these up? You just want me to do this? Okay. Then we can do this, and then... Okay. So now, we'll go ahead and go to this side. Okay? So if we have adenine here, what's bonding to adenine? Okay, to guanine. To guanine. And then to thymine. Okay, next. 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 And, okay. So this is our double helical shape. We said it's like we tip this over like chopping a tree down, which is what we're going to do. Okay, we don't need these anymore. Well, we do, but then we open up what's called a replication fork, and it looked like this. Okay? So then what we need to do is draw these in. So these go by 3s. 1, 2, 3. 1, 2, 3. Then 1, 2, 3. So we'll have 9. Yep. Okay. So we just start from the front and the top. So we do AGG. Okay. Then we do TAC. And then GGA. Okay. Then we'll put then we can put these complementary base pairs in. Okay? We can just look at this and just know that TCC and then ATG and then CCT. Okay. What happens is, notice it said something about an enzyme. Okay, so I'm going to put these little dots in here, just 3 of them, because this is what we call hydrogen bonding, because it's hydrogen atoms that hold this together. Okay, kind of off at a slant there, but we get the idea. So we just have this circle, and this circle is just an enzyme moving in this direction. And it's what we call DNA helicase. Okay. So, So now, do you want to try yourself putting these letters on the outside bottom and outside top, or you want to do that together? So now's your chance to participate or whichever, so however you want to look at that. And then when tomorrow rolls around, okay, I know what we're doing. Okay, I'm asking for input. You can do them? Okay. And then I'll go through these and then see if we get the same results. So I'll just hit pause for just a moment. Okay, so we've taken a little bit of time. Now we can see what we have on here. And notice that, again, adenine bonds to thymine, cytosine bonds to guanine. You don't even have to look at these as you go by. You can just read the bottom here and just say, I'm willing to bet that from right to left, it says TCC, ATG, CCT. Looks right to me. Provided if it's done correctly on the inside, then it's got to be right on the outside. So the red letter should be AGG, TAC, GGA. AGG, TAC, GGA. Exact same thing. So then what happens if you replicate something? You got 2 shots and a free throw, or you got 2 shots to shoot free throws. You make the first one. What happens if you replicate that? You make the second one. So that's what we're trying to do. In other words, you're trying to copy that. That's exactly what we have or are seeing take place here. Then, on our last step, okay, we're going to describe all this in the book on how this is actually happening. There's 2 more enzymes now. This one's called DNA polymerase now. I'm going to run out of space. And there's one down here too, moving in this direction. DNA polymerase. Okay, so I would recommend that you write that in. Okay, then once you've done that, just open up to page 333. So number 3 and it appears 3 times. What a coinky dink, huh? So in the blue, on page 333, about a quarter of the way down, what does it say? Semi-something.
Conservative replication.
Yeah, semi-conservative replication. Okay, so just follow along, page 333. When Watson and Crick presented their model of DNA to the science community, they also suggested a possible method of replication called semi-conservative replication. During semi-conservative replication, parental strands of DNA separate, serve as templates, and produce DNA molecules that have 1 strand of parental DNA and 1 strand of new DNA. Recall that DNA replication occurs during interphase of mitosis and meiosis. An overview of semi-conservative replication, figure 10, okay, which we can look at here in just a moment. The process of semi-conservative replication occurs in 3 main stages: unwinding, base pairing, and joining. So the first process, unwinding, this DNA helicase is causing this to be unwound and it breaks it apart. That's the function of this DNA helicase. Okay? Where is this process taking place? At the ribosome, at the lysosome? Neither one of those is correct. Where did we say cells get their instructions from? So this must be taking place in the nucleus, which it is. Okay? So once these bonds have been broken, what were these three dots? What did we say that they were? It starts with an H. Nope, I mean, that does too, but these are hydrogen bonds in here. That this DNA helicase breaks these apart, and now you see this fork opening up, okay? Then these nucleotides, whichever order they're specified in, just start to line up with its corresponding partner, if you want to call it that. Okay, so it comes unwound, base pairing takes place, and then it gets sealed shut again. That's what that last sentence had said. It said, the process of semi-conservative replication occurs in three main stages. Unwinding, here's being unwound, DNA helicases breaking it up. Base pairing, that's what we did down here. And then finally the last one is joining. That means that this DNA polymerase is sealing this new strand up. Because if this was to keep going and keep going and keep going, which it does, all these hydrogen bonds just get broken. I think it's fair to say that now, what do we see? If this is not joining together anymore, how many strands of DNA do we have? We started with one, now we've got, so you've now replicated some directions in the nucleus. Now you've got two strands of DNA instead of one. Okay. So, as we continue on page 333, DNA helicase, an enzyme, is responsible for unwinding and unzipping the double helix. That's what we see happening here. Okay? When the double helix is unzipped, the hydrogen bonds between the base pairs are broken, leaving single strands of DNA. Then proteins called single-stranded binding proteins associate with the DNA to keep the strands separate during replication. As the helix unwinds, another enzyme, RNA polymerase, adds a short segment of RNA called RNA primer on each strand of DNA. Okay? We're not too concerned about that. Base pairing, that goes back to adenine and thymine, cytosine and guanine. That's how those two bond together. The enzyme DNA polymerase catalyzes the addition of appropriate nucleotides to the new DNA strand. The nucleotides are added to, okay, we're not too concerned about 3' and 5'. DNA polymerase continues adding the new DNA molecule to the chain by adding to the 3' end of the DNA strand. Recall that each base binds only to its complement. A bonds to T, C bonds to G. In this way, when the templates allow identical copies of the original double-stranded DNA to be produced. So the main idea, we're thinking about four things here. The first one, DNA breaks these hydrogen bonds and opens up this fork. That's step one. Step two, we have all of these nucleotides that are floating freely in the nucleus start binding up and matching to their complementary base pair. And then we have DNA polymerase that is sealing this back together again to where it binds these to form a new DNA strand. I said 4 phases. We'll just say those 3. Step 1, these start to match up in step 2, and then step 3, this DNA polymerase seals that back together to go from 1 strand to 2. Okay. Now, it says transcription today. Okay, move to page 337. Is there any questions on this before we move forward? Front row, are we on the bus here? Middle row. Back row. Back row. Back row. Back row. Okay. So, on page 337, transcription. The first step in the central dogma just means something that we don't understand. Okay, the first step in this involves a synthesis of messenger RNA from DNA in a process called transcription. The way that I like to describe this is when you look at a scribe back in medieval times, and that's how I describe it. What do you suppose a scribe could do in this feudal system where you had peasants, you had scribes, and you had a knight who probably owned the land here in a kingdom? But what could scribes possibly do? Do we know? They're not just individuals who could write. They could also do what? Probably. If you can write, you can certainly do this as well. It's what we're doing right now.
Learning.
We're learning too. Reading. So scribes are individuals who could read and write. The reason we say scribes is because we're talking about a script or transcription. Okay? So the synthesis of messenger RNA from DNA in a process called transcription. Through transcription, the DNA code is transferred to a messenger RNA in the nucleus. The messenger RNA then can take the code into the cytoplasm for protein synthesis. Follow along with the process of transcription. The DNA is unzipped, much like what we see here in replication. Transcription and replication are two totally different processes. Okay? The DNA is unzipped. The nucleus and RNA polymerase now, an enzyme that regulates RNA synthesis, binds to a specific section where the messenger RNA will be synthesized. As the DNA strand unwinds, the RNA polymerase initiates messenger RNA synthesis and moves along the DNA strand in the 3 to 5 direction. The strand of DNA that is read, see, we're talking about scribes, could read and write. Okay? So the strand of DNA that is read by RNA polymerase is called the template. Okay? Template strand. And mRNA is synthesized as a component in the DNA nucleotides. The DNA strand is not used as a template strand called the non-template strand. The messenger RNA transcript is manufactured in a specific direction, adding each new RNA nucleotide to a specific end. Uracil is incorporated instead of thymine. As the messenger RNA molecule is made, eventually the messenger RNA is released and the RNA polymerase detaches from the DNA. Okay? Now, it says the messenger RNA then moves out of the nucleus through the nuclear pores into the cytoplasm. So if this was going to make a protein, which it does, what is it, or where does protein synthesis take place? It's on this structure on the endoplasmic reticulum. It starts with an R. Nope, it's not RNA. From last chapter, 2 chapters ago.
Respiratory.
Nope, not respiratory. The ribosome. So this is heading towards the ribosome where proteins are then going to be assembled. Okay, so on page 338, okay, this is what we're talking about here. What's going to happen is now we're going to read the outside of this bottom strand here. But if we have an adenine, we don't bond to thymine. We bond to this one instead, and it starts with a U. It's called uracil. Okay? So wherever we see an A, we would put a U. Okay? So if that's the case, if this was DNA, we would put a T. But now we're doing mRNA. Okay? So what bonds to adenine? Uracil. Same thing is for everything else. What bonds to guanine? Well, what bonds to guanine? Cytosine. What bonds to guanine? Still cytosine. What bonds to thymine? Now this is DNA still. Okay, so maybe I should put this down just a little bit so that we know that the bottom is messenger RNA and the top is still DNA. Okay, so now what bonds to thymine? What bonds to thymine? Adenine. What bonds to adenine? What bonded to this adenine? U or uracil. What bonds to cytosine? Guanine. What bonds to guanine? Cytosine. What bonds to guanine? What bonds to adenine? Uracil. Okay. So, these are specific amino acids. They come in 3-letter pairs. Okay? So, on page 338, it's got this deal that looks like this. It's a table. Looks like this. Okay? Table or figure 14. We want to show you how you read this now. Okay? So, the first base. Okay? You just go, here it says first base. These are your first 4 choices. Then you go across the top for the second base. Then you go across the bottom for the third base. Okay? So, if our first base is uracil, okay, what color is uracil? It's red. So we're somewhere up on the top here. So we're at uracil. What is our second one? Okay, so then in the red, we go across till we find C, and that's what color? Okay, then what's our third level, or letter? Still cytosine. So in the red, okay, then the blue, then we go down till we see the blue again. So what amino acid is that? And it would say UCC. Serine. So these codons, that's one of your vocab terms too, these codons are coding for this specific amino acid called serine. Okay? Now, what's going to happen is as we're going forward, you will be responsible for completing this. We're doing an activity on it, you're given this table, and you have to come up with the proper amino acids in this sequence. Okay, do you want to take a stab on what amino acid this is coding for? Or do you want to work on it together? Okay, so what is, now you read it going this direction from right to left, not left to right, because it's going in that direction. So you got to do first, second, third. And that is a very important amino acid. Methionine. It says something very specific besides AUG, besides methionine, it says something very specific as well. It's a start codon. So really, what happens is typically this should be up front. This RNA polymerase is looking for this amino acid or this code for this amino acid. So RNA polymerase knows, okay, we've got methionine. That's where this process has to take place. Okay, we're running out of time. So tomorrow, what you're going to do is make models of DNA replication and transcription. Okay, but I will give you time to collect your thoughts because this might have been a lot, but otherwise moving forward, back row. Are we good? Back row. back row, back row, back row, back row, back row. Okay? Back row, back row. Okay? Middle row, front row. All right. We will catch up to you next time.